Alrighty. I need all eyes paying attention. We are going to be discussing some things as well as taking notes to get a better understanding. You should have this page already labeled and documented as page 35. And as we're going through, you are going to assist me with making sure that we get these notes taken correctly. Let me move this thing off the screen. Can everyone see this? Everyone good? All righty. So then let's go ahead and take a look at this information. All right. So what we know so far is the cell. We didn't talk about the second portion. Actually, you did talk about it a little bit when you did the webcast and you were moving through stations and you were getting some information about the cell membrane. But one of the particular things that we actually talked about with the cell membrane is we know that it's mainly made up of that what? Not the cell membrane. Where's the cell membrane located? On the outside. And we built a model. What's that outside made up of?
Hydrophilic and
What are those called? Phospholipid. So we know that they are made up of phospholipids, but they're also made up of, who remembers from looking at the little interactive activities that you did where you had to be able to build the cell membrane and let certain things in and not let certain things in, but there were other things that you put that made the cell membrane. Also starts with the P, and it's a macromolecule.
protein.
Protein, very good, Jenny. So our cell membrane is made up of phospholipids and protein. So let's go ahead and draw this cell membrane in this space here. So Rosalind told us that it's made up of what? And I put colored pencils there in case you want to do your notes in color. But it's made up of a what? And what is the phospholipid made up of? Hydrophilic what? Heads and hydrophobic tails. So leave a little bit of space above here because we're going to write something there. So we're going to start it right about here. I'm actually going to draw this in pencil only because my marker's a little too thick. So we know that the heads are hydrophilic, which means that they are what? They're water-loving. So I'm going to draw a little heart. It's hard to see it on the screen, but I'm drawing a little heart there. Is it made of just one pair? No, many pairs. So we should draw a couple. And again, I'm putting a heart to signify that they're water-loving. But not only is it made up of phospholipids, we also said that it's made up of what? All right, so before we do that, we want to label these to be what? Phospholipids. And the phospholipids contain two lipids, so we call it a what? Bilayer. So if we put over here, phospholipids form two layers, which is a bilayer. So we want to label this the bilayer. One other thing I want to point out as well, I left a lot of space on the left-hand side because throughout our notes, we'll be jotting down some extra information to help us understand our notes. So that's why I left that space there. Remember, I told you I was going to be doing that. All right, before we enter our other piece of our membrane, this portion out here would be considered the inner part of the cell or the outer part of the cell? This is outer. This would be the outer environment. That's outside of the cell. Does it contain water? Yes. Which is why the hydrophilic heads are there, so we can put that it's watery. And I'm just going to go ahead and add another phospholipid just for the sake of space so that when I'm drawing next, will be in its proper spot. What is this location here? This space here? This is inside the cell. So that would be considered the what inside the cell? What's the liquids, the fluid inside the cell? Cytoplasm. Is that watery?
No.
Yes.
Yes.
Remember our motto, that jello, that's a liquidy. All right, so we have that our cell membrane is made up of phospholipids, bilayer, but it's also made up of what? Proteins. So let's go ahead and draw a protein, and we'll just signify it by a big block, and we'll label it a protein. And we'll continue by just putting maybe another set or two of phospholipids. So that's a picture of our cell membrane containing what? Phospholipids and proteins. Now when studying your notes, how do you think you're going to study? How do you think you're going to study? The picture will help you, but how do you study it? Just looking at the picture is not studying. Remember, you have to, just like you guys were talking about sports today, when you work a muscle, it will be sore because you have to work it, but you then have to make it work. Your brain is a muscle. How do you make your brain work to study this? Just reading it is not studying.
Analyzing.
How do you make your brain remember what should go here? How?
Like you can draw it and try to label it.
Where is there a blank copy?
On your website.
Shouldn't you go to the blank copy and see if you can fill in these blanks
Yes.
and draw it? And where's your answer key?
Right.
Your notes. And I've been saying this to you guys year after year. If you do this, you will be successful. If you haven't been doing it and you weren't successful, then don't be insane and not do it. All right. So moving over here, we have that our phospholipids are two layers. It's a bilayer. It gives the membrane the ability to be what? When you were actually making your bubbles had the ability to be how? It didn't pop, okay, so therefore, was it weak or was it strong? So it gives your cell membrane strength. Now, Jalil did something very interesting with his bubble. He kind of moved his bubble around. And the fact that the bubble was able to move a little bit from one location to another signified that the bubble was kind of what? Maybe starts with an F. Flexible. So because the cell membrane is made up of this two-layered phospholipids, it allows it to be strong so that things can come through and go out without breaking the cell membrane, because if we break the cell membrane, what's going to happen to the cell? It's going to be damaged. It also allows it to be flexible to allow certain things to go in and out of the cell. Any questions with that? Alrighty. Now, I want you to stand up out of your seat for a second. And I want you to demonstrate fluid to me. If I said be fluid, how would everybody should be up? Everybody. Everybody up out of your seat. How do you demonstrate fluid? Everybody, demonstrate fluid to me. I don't need you to talk. I need you to demonstrate it. All right, go ahead and have a seat. Yes. Now, what was it to be fluid?
To be free and
Freely moving. Okay, so you all were kind of moving around to be free moving. Now, the cell membrane is a fluid structure. What does that mean? It moves freely. Let's see if we can fill in the blanks that's following after. Take a look at it, read it yourself, and see if you can kind of maybe fill in the blanks there. Raji. So the cell membrane is a fluid structure, which means the phospholipid bilayers of the cell membrane are able to move because that hold fluidity, that fluid means that it's able to move, and they actually kind of move side to side. They flow, and sometimes they actually trade places with one another. And that's what allows that cell to be able, that membrane to be able to allow things to go in and out. And the fact that they're able to move side to side, switch places, says that the cell membrane is also what? If I tell you, Brennan, do you mind switching places with somebody? And Brennan says, yeah, that's no problem. Then Brennan's not being stubborn. He's actually being pretty, what's the F word? Flexible. Not the other F word. Did we recognize that in our bubble structure? Yeah. Yes, we didn't see the phospholipids, but we did see the fact that it was kind of a fluid structure. It allowed things to go in and out, so that's that flexibility, and it was strong without breaking or popping. Any questions about that? All right, then let's go ahead and look at the job function. What is the job function? Write down right now in your own words the job function of the cell membrane. Write it in your own words right now. Nope, I just want you to write it down first. The job function of the cell membrane. All right. What did you have to do to actually allow your object to actually pass through? What object actually passed through the cell membrane? In your the straw. What did you have to do? You had to put some of the solution on it to allow it to pass through. Did the toothpick pass through? Why? Because it didn't have any solution on it. Okay, so what's the job of the cell membrane? Did everyone get that part right? Yes. All right. So the fact that the straw did go through, but the toothpick didn't, does the cell allow everything to pass through? No. No, only what? Some. What's that? Only allowing some things. Is it all the way allowing or only some way allowing? What's a word you can think of that would say not all, but some? Another word starts with an S. Like think of a circle. It's not all of the circle. It's only half of a circle. Semicircle. So therefore, the cell membrane is semi-something. Now think back to 8th grade, if you will. This is that passing through of substances. So remember, who said it? Permeable. Permeable. Very good. Awesome. I know I taught you something. So the cell membrane is semi-permeable. And that means what? It only allows certain things. And when I say things, I actually want to put the word molecules. It only allows certain molecules to pass through. And remember, if this were not a bubble that we were actually doing, if the toothpick would break the bubble, if this were actually a cell membrane, would the toothpick be allowed in? No. So it only allows certain things to pass through and prevents. Kind of like how Aaron stopped Keelan from getting in yesterday. Prevents other substances from entering. The cell. Shauna, did I provide enough space for you, babe? Did I? Good. Any questions? All right, so the cell membrane is semi-permeable. What we also said, the cell membrane is a what for the cell? If it's kind of, it's a barrier. Good, it's a barrier. And it's a barrier not of all things, only for some things. So it's a selective barrier. So you can identify the cell membrane as being two things. Either it's what? Look at these notes. Either it's going to be what? The two things both start with an S. Semi-permeable or selective barrier. They both mean, what does that mean? They both mean what? Only allows certain things to enter and prevents certain things not to enter. Any questions for that? All right, I'll give you a moment to get caught up with writing. All right, when you push the straw through your cell membrane, did the cell break? No. But did there have to be an entrance made for that straw to get in there? But the cell did not break, so therefore we know the cell membrane is capable of doing what for itself? But once you took the straw out, was that opening still there? No, so the cell. So that means it's able to do what for itself? Heal. So self-repair. Good job, bestie. So we had that substance had to break through, but it didn't break the cell. So the cell membrane is capable of self-repair. Let it in, but it didn't damage the cell. Any questions for that? All right, so going back again, what did we have to do in order to allow the straw to go in? We had to put solution on it. So therefore, the straw needed guidance. It needed something to be able to enter. It kind of needed a ticket to get in. It just couldn't get in by itself. It needed an assistant. That's kind of like if you went to a concert and you were trying to get backstage to meet who's coming. No, who's coming? Who's coming next? Who did my son say was coming next week? No, Schoolboy Q is coming, right? Okay, so Schoolboy Q is coming, or Rihanna's coming, whatever, is coming for a concert, but you want to get backstage. You have to have what? You got to have a pass. You got to have that VIP. That VIP is going to let you get past the guards to get through. If you don't have the VIP, the guards are going to do what? Nope, you're not getting in here. You're not getting in here. So the straw needed a VIP. It needed a pass to get through. So proteins and cell membranes, these proteins are the VIP. So if you want to put over here, they're the VIP for entrance. The proteins are the VIP for entrance. And they're actually called transport proteins. Now the proteins are the passageway. The passageway word that we use is channel. So they're the VIP for entrance. They're going to be called transfer proteins. They're going to be the passageway or the channel that's going to help molecules do what? Not just travel, but get in. They're going to help molecules actually pass through. This process, having a molecule or having something assist to get through a cell membrane, this is actually called facilitated diffusion. And we're going to talk about the word diffusion in a moment. Hold the thought because I'm going to ask you in a second. Any questions for this? All right, so if we're talking right now about this protein, so the reason why I left this blank is because I didn't want to give you a hint to what went up there. But if the transport protein, if a transport protein that is responsible for assisting passage for a substance like glucose, because glucose has to enter the cell, is not functioning, then the membrane is going to do what? Now think about it this way. If you are going to the concert and you're VIP, what